Thank you very much. Our next speaker is Deb Money, and Deb is from BC Children's Women's Hospital and Health Center, and is speaking to us on the vaginal microbiome and health and disease. Please join me in welcoming Deb.
So thanks very much for inviting me. It's always a pleasure to be here at one of your symposiums. And I'm going to kind of continue on the human microbiome theme and talk a little bit about the work we've been doing trying to study the vaginal microbiome, a very important aspect of human health for 50% of the population at least. And we're really grateful for the funding that we've had from CIHR and GNBC and others. So why do we care about the vaginal microbiome? Well, as in the gut and in other niches in the body, the vaginal microbiome is that balance of microorganisms that really can be quite fragile at times, but the nature of that balance is really associated with the ability to defend against pathogens, and also an imbalance or a dysbiosis in this area is associated with serious health consequences, both for the woman from the perspective of post-surgical complications. One of the most common reasons for attending a physician is actually related to vaginitis and vaginosis, but also, very importantly, adverse perinatal outcomes. So a major cause of unexplained prematurity. So our VOGUE study, which is our vaginal microbiome group initiative that's funded by the Canadian Microbiome, or part of the Canadian Microbiome Initiative, is actually a collaborative between a number of sites in Canada. And our approach has been to enroll a series of different clinical cohorts to try and answer the question of what defines health in this area, and also what are the markers for disease. And to just give you a sense, and I'm going to show you just a sampling of data from a couple of these cohorts. We've been collecting samples from healthy non-pregnant women, samples from HIV-positive individuals, non-pregnant women with recurrent vaginitis, low-risk pregnant women, low risk for prematurity, and women at high risk of prematurity. And we're using a CPN60 gene target because of the ability in what's actually a rather distinct microbiome with a high predominance of lactobacilli. The ability of CPN60 to discriminate for us has actually been very helpful. We use 16S RNA in some cases for quantification, and I'm not going to show this data, but we're also doing some transcriptomic, virome, and metabolomic work, and we're working with a social scientist to understand the behaviors that are associated with changes in microbial patterning. So I'm just going to give you some highlights of data from three of our cohorts. One that's not part of the VOGUE group, but we've had funding from March of Dimes to look at women with preterm premature rupture of membranes. But we've got our healthy cohort who are 18 to 49 years old, HIV negative, not using antimicrobials, pretty healthy, not recently disturbed microbiome. We've taken a very healthy group of pregnant women and sampled them, and then we have these women who have the misfortune to have spontaneous rupture of membranes between 24 and 34, just about 34 weeks gestational age, and we've been following them. Our methodology is not dissimilar to many microbiome approaches, where we take our sample, which is simply a swab, extract DNA, use our CPN60 target, amplify it, and then use parasequencing for our sequencing methodology. We put it through a software pipeline that is extraordinarily complex, and we've been changing how we've done that over time a little bit. And then the assembly method has also varied a little bit over time, but we end up with outputs of our operational taxonomic units that we've married to the CPN60 database. We've used a slightly different assembly more recently, where we've actually tried to assemble a number of our cohorts to get a reference assembly and then compare that. And that's altered a little bit of how we've understood our profiles. So first off, just to give you a sense of our healthy cohort, this is just a list of the organisms we've been able to identify, at least as best as we can, to the database. where you see Lactobacillus species or genus dominating, but a number of subspecies of Gardnerella vaginalis, which is a very important organism in the vagina, and Prevotella really coming out as a very significant organism, and we'll talk a little bit about that in a minute. What we've been able to do, and this is a shifting landscape with our other groups around the world trying to understand community state types. With our methodology, we've come up with 6 primarily that everything sort of shakes down into, which is different Lactobacillus dominant groups and then different sub-Gardnerella vaginalis dominant groups. And then 2 that really are very diverse, which one is just a huge diversity, and another diverse group that seems to have one sort of alternate organism dominant, like a bifido, a dominant or Prevotella dominant group. And those tend to be seen more in our situations of dysbiosis. So this is just another way of us having to look at our data, where you see our organism types down listed there. It doesn't matter which ones, but you can see the patterning. And red, of course, in these heat maps is higher proportion. And then over the top, we've overlaid our community state types and then also gram stain categorization. And so this is just to sort of demonstrate how we're trying to visualize our data. So then we moved into looking at the vaginal microbiome in pregnancy and asking the question of what does the microbiome look like in somebody who's very low risk, who presumably is going to have a completely healthy term outcome, is pregnancy itself something that changes the microbiome, or is it similar to non-pregnant? And we really need to answer that very, very simplistic question before we went on to understand any abnormalities. And so these are, again, just some samples of what our pregnant women look like. So we had just over 300 women, and the vast majority of them actually looked quite homogeneous, where they had huge numbers of them were just Lactobacillus, primarily crispatus, a little bit of Lactobacillus jansenii, and then a few with Prevotella and Gardnerella, but really primarily looking quite undisturbed, and then a very small number, just 25 out of our 300, that had this really diverse microbiome. And we're still looking at clinical outcomes to see if, in fact, these low-risk healthy women with these profiles may not have entirely normal outcomes or not. The other thing we're interested in looking at are molecules. So because molecules don't have a CPM60 gene, we've had to use targeted PCR to look at these. And we're interested in mycoplasma and ureaplasma in the pregnancy context. And we find there is a difference in pattern between pregnant and non-pregnant. We're not sure what to make of this yet, but at least we're understanding what the shift is. So basically, we've concluded, at least so far, that the microbiome in healthy pregnant and non-pregnant don't look particularly different, except that really low-risk pregnant women actually have very low diversity and low richness in their microbiome. But there is a difference in the prevalence of molecules. So then just to very quickly touch on prematurity. So the outcome of a preterm delivery is really an obstetrical disaster, because although our neonatal intensive care colleagues are outstanding at resuscitating and having these little babies, this is an infant with a pen to give you a sense of size of this premature infant. The outcome can be really tragic with significant developmental delay. It's also very, very costly, 1 to 3 billion per year in Canada, and accounts for 75% of neonatal morbidity and mortality. So if we could get to the answer of this would really be a holy grail. And our basic understanding is that organisms that reside in the lower genital tract and the vagina actually ascend and cause weakening due to bacterial byproducts of the membrane and cause this inappropriate rupture or trigger inappropriate contractions much too early. And the trade-off clinically is that as we leave an infant in utero, the risk of infection and inflammation rises, and if we deliver early, then we have neonatal morbidity and mortality. So we're beginning to try and pattern the microbiome at the time of rupture and looking at some clinical characteristics and seeing that we're having a very, very diverse microbiome in this population. We're seeing Megastera and Prevotella being very dominant in this preterm rupture group, which is really not seen so much in our low-risk pregnant. And we're seeing molecules being significantly different, and these are individuals that delivered early versus late, and individuals who had lower gestational age versus higher, and lower birth weight versus higher, and having higher proportion of molecules present was associated with worse outcome. So really this is just a little snapshot of some of the data we're seeing, but I think this visually shows it the best. That on the far right for you is our low-risk pregnant group that just look really homogeneous and really, from our perspective, super healthy with lots of lovely lactose. Our clinically healthy non-pregnant, a little bit more diversity, but still very lactobacillus dominant. And the preterm rupture of membranes profiles are just all over the map with great diversity and multiple types that we would consider more of our pathogenic microorganisms. So this is just a beginning of us trying to understand where we're at with regards to health and disease from the perspective of this microbial niche. And as Stuart alluded to, I sure as heck don't do this alone, and I have a huge team of individuals, both here in Vancouver and as extraordinary partners in veterinary science and Agriculture Canada, actually in Saskatchewan, as well as colleagues in Toronto, London, and Guelph that have really contributed to many aspects of this project. So thank you very much.